Hi, everyone. Welcome to the Spatial Omics Seminar Series 7. And today we are very happy to have 10x Genomics presenting their multi-omics work. And we have Dr. Marlon Stoeckius from 10x and located in Stockholm, Sweden. And briefly, Dr. Stoeckius received his degrees, the PhD degree from actually Germany in Max Delbrück Center in Berlin, Germany. Then after 2 postdoc experiences in Berlin Institute for Medical Systems Biology and Yale School of Medicine, he joined New York Genome Center as a research scientist where he worked on development of next-gen library sequencing for Illumina platforms. Then around 2019, during the pandemic, he joined 10x Genomics to lead the efforts of spatial transcriptomics technology development. And recently, he also has been performing the role of co-director of R&D in Stockholm at 10x Genomics to further develop the spatial omics products integrating proteomics and genomics. And without further ado, I'd like to turn over to Dr. Stoeckius.
Cool, thanks so much. Let me just quickly share my screen. Yeah, I mean, I just first of all, I want to thank for the possibility to speak here. I think the seminar series has really been amazing. I think through the whole pandemic. I mean, a lot of us were always looking forward for the next Friday to hear some talks because we were all aching to go somewhere else and see some interesting talks, which was obviously not possible for a long time. So I really hope you guys keep this going. I think that's really great. So again, I mean, I'm Marlon Stoeckius. I'm co-director of R&D in Stockholm, which is really our spatial technologies R&D hub for 10x. We're right in the heart of Stockholm in a new up and coming sort of medtech hub right next to Karolinska Institute and the SciLife labs. So I'll talk about the technology today that probably isn't new to most of you guys, but I just want to get a quick intro so that we're on the same page. So this was originally actually developed here in Stockholm, and it's called Spatial Transcriptomics or Visium. And what this allows you to do is take a tissue section, either FFPE or fresh frozen, and take an image of it under the microscope, either by IF or immunohistochemistry. And sorry, not immunohistochemistry, by immunofluorescence or H&E staining. And then you can make an unbiased whole transcriptome library out of this tissue section at high spatial resolution. And I'm just going to exemplify this here with a mouse kidney, which is nice, to work with because, maybe you've seen a lot of work on the brain from us. We typically work with organs that are very well studied, where where things should be expressed. And, if you get the data back, that makes sense or not. So the first thing what we get is this H&E image, and then the next data point is this whole transcriptome library at high spatial resolution throughout the entire tissue section, and that's essentially seen here in these spots that are highlighted, and the different colors mean basically an intensity of transcripts that were sequenced there. And I'll talk a little bit later about where these spots are actually coming from. But one of the first things that we typically then do with these sorts of data, you just treat all those measurements as sort of individual measurements, and you can cluster these, and sort of the same way you're used to looking at in a single cell, and these typically cluster up into areas that are transcriptionally similar and that have oftentimes very same biological functions. And then what you can do is you can cluster, you can basically project these clusters back onto the XY coordinates where these were coming from, and then typically what you see is that regions that are functionally similar, or like structures that you maybe know in this case in the kidney, will cluster together. So for example, in the cortex, you see that there are 2 different types of clusters, and then the medulla, inner medulla, and then I guess this place is called the pelvis of the kidney. But of course, I mean, besides these whole transcriptome measurements, you cannot look at individual marker genes, and it's just highlighting a few genes that are specific for these different clusters and these different regions. You see that it's very nice spatial expression patterns of these different marker genes, from the cortex into the medulla and into the pelvis. So this is essentially a high-level overview of the workflow. So what you do is you section a fresh frozen tissue section or FFPE tissue onto these glass slides, and I'll show them in the next slide a little bit more in detail. You do the imaging, so either immunofluorescence or H&E, and then you do a library prep, partially on the slide and then also in a tube, and you submit those libraries for sequencing. And after that, when all the data comes back, you can analyze the data with the tools that we also provide. So this is essentially a platform, a tool that allows you to collapse the imaging data together with the sequencing data. And then the Loop browser is a desktop application, easy to use desktop application that you can to visualize your data on and then browse the data on a standard desktop computer. So if you have not any experience with computational biology, this is really a way to then explore your data and get some first insights how it looks like. So the core of the technology, and that's essentially what we got originally developed here in Sweden, is this slide that looks like a standard glass slide and has 4 capture areas. And these capture areas are 6.5 by 6.5 millimeter. And on the surface of these capture areas are these 5,000 barcoded spots. And these are essentially 55 micron diameter lawns of reverse transcription primers. And if you're used to looking at the primers that we have in our single cell beads, and this will be very familiar to you. So these have a partial read 1 primer that allows you to amplify this and also then sequence it at the end of the day.
That was pretty much it. So basically
this is spatial barcode. That is essentially, from single cells, it's a single cell identifier, but in this case, it's actually XY coordinates on that surface. There's UMI for counting the transcripts, and then there's a handle that captures the mRNA molecules. And any mRNA molecule that gets captured here and gets reverse transcribed and indexed essentially with this barcode, when you do the sequencing and you then do the data analysis, you can collapse it back onto the XY coordinates, and you know where this transcript was essentially coming from. So that's the final library, how it looks like. And the structure is very similar in our single cell and our Visium product. So and the only thing that's essentially different is the barcode. And for single cell, this barcode is referring to a single cell. And for spatial, this barcode is referring to a spatial barcode. And then you can treat these measurements essentially like individual measurements and map them like this, or you can map them back onto the XY coordinates. So the kit has been used by various researchers and in many different tissues and disease areas and species. So for example, human cortex, lung, and mouse, a lot of mouse tissues. But when we saw more and more data coming for the tumors, we realized, this was actually originally a fresh frozen kit. I should have mentioned this. When we saw more data coming out on tumors, we realized one of the most promising, most prominent samples that are out there or most widely available is actually FFPE. So that's why we developed this FFPE kit that was essentially described, I think, by James last year in the same seminar series here. And interestingly, so the first paper actually came out of a lab and looking at and studying the tertiary lymphoid structures in renal carcinomas using the FFPE kit, but also using the fresh frozen kit. And so what they did here is basically they wanted to study immune cells and how these immune cells are organized in these tertiary lymphoid structures and which cells are also in there. And they basically realized this in a lot of B cell infiltrates and also some T cells. But one thing that became evident, and that was something that we've also seen before, is some immune cells or many immune cells are actually very difficult to characterize based on RNA alone. And one example here is so the cytotoxic T cells would have been really difficult actually to identify where they really are within this tumor. And that's because many immune cells, first of all, they're very small, but then also many very characteristic markers for immune cells are oftentimes slowly expressed, and they can be also post-transcriptionally regulated. So this one example here, where a resting T cell, gets activated over TCR signaling, and then of course it starts to produce proteins that were before translationally silenced. So these two cells look very similar on RNA level, but on protein, they're extremely different. And then, of course, this cell is activated. So there's a lot of signaling cascades, a lot of post-translational modifications that are ongoing that you don't really see in RNA. And then, of course, there are a lot of other global modifications that are used to characterize tumors as well. So, for example, histone modifications. So if you have hypomethylation of certain marks, it's a hallmark of tumors, and that's oftentimes used. And then one example that I oftentimes also give for looking at RNA versus protein is isoforms. So mostly we don't look at isoforms so much on the RNA level. But on protein, for example, the marker CD45 RANRO, which distinguishes memory from naive cells, is a very prominent marker, obviously, that's been used a lot in with antibodies. And then, of course, if you talk to pathologists, they're oftentimes used to looking at protein to characterize cell types and cell states, and not so much used to thinking about RNA. So if you want to traditionally type a cell, it's oftentimes done with proteins. So that's why we wanted to essentially develop a kit that allows you to do RNA and protein in parallel from the same tissue section and from FFPE in this case. And so the kit will be coming out in the middle of the year, and I'll talk a little bit more about this, but I just want to give you a quick highlight. So this will come out with a panel. So that's very different compared to what we've done before. So there will be a validated panel within the kit. And this will contain intra and extracellular markers. And you will be able to do H&E staining or IF staining on top of this. So the method essentially, so I'll quickly introduce you how this works, and want to take a step back and actually quickly also introduce how the RNA detection works in this case. And that's something that James talked about last year, but I just want to see that we're all on the same page here. So obviously in FFPE, the RNA is highly degraded, and it's pretty difficult to make a truly unbiased RNA expression library by pulling out the mRNA. So what we developed essentially is this method that we call RNA Templated Ligation, where you have two probes for a target mRNA, but in this case, these are probes essentially for the entire transcriptome, so that's as unbiased as possible. And only if these two probes are binding to the RNA molecule of the target molecule, they get ligated there, and only then they make a constructive, constructive molecule that can actually get sequenced. So one end has a poly-A tail for the capture, and the other hand has a handle for making a sequencing library out of it. And then when you digest the RNA and the tissue, these molecules then similarly to the mRNA molecules in the standard assay diffuse to the surface where they get barcoded. For the protein detection side, that we use these DNA-barcoded antibodies, and that's something you've maybe also seen for single cell before. So these antibodies are tagged with an oligo that contains a true seq-re2 handle that is used for sequencing and also for amplification. Then there's a 15 nucleotide barcode, which is essentially the identifier sequence of this antibody. And it's unique for essentially every antibody. And then you have a capture sequence that allows you to capture this molecule on the surface of our slides. And if you think about it, it's a DNA barcode in this case that identifies the antibody. There are much more multiplexing capabilities here. There are much more, you can imagine much more different sequences here than what's available for flow cytometry or for CyTOF. So the way the assay works is you have these probes, these transcriptome-wide probes, and you have these antibody panel, and you throw it onto your FFPE tissue section. And then within the tissue section, the antibodies will bind either to intra or extracellular markers, depending on which antibodies you're using. And then these probes will bind to the RNA molecule of interest. And then you dissolve the tissue essentially, and the RNA molecule, these RTL probes, and the antibody oligos diffuse to the surface where they get captured and indexed. And that's what you see here a little bit in a blow up. So these are these transcriptome-wide RNA probes. They bind to the surface, and the antibody oligos bind to the surface where they get extended. So they get barcoded, so you know where they are coming from. Essentially, then you release them off from the surface. And then the first thing that you do is you make a library for these two products together, a pre-amplification. And then after this, you split this into two individual tubes where you then amplify a gene expression library and the protein expression library independently. And then you can sequence them together at the end of the day if you want to, or you can also tune then the sequencing depth for each of these two, depending on what you want to know a little bit more about. So we essentially wanted to develop an assay that for immune markers, and then of course you need to work with a tissue that's studied very well. And I guess you've seen data from us earlier in the time where we always use the mouse brain, because really what you're looking at. There's a lot of data that's available for the mouse brain that you can compare to. So when you work with when you want to develop an immune panel, obviously you can't use the mouse brain. So you use typically other organs, immune organs that have very highly repetitive or well-described structures that you know very well, and when you get the data back, if an assay worked or not. So this could be, for example, the tonsil, but it could be also the spleen or the lymph node. And here's an example for the tonsil, where you see very nicely these macroscopic large structures, which are they're called the follicles that contain maturing B cells, and then surrounding those as a layer of T cells and monocytes and other immune cells. And this is again in a blow up here for immunofluorescence image for different markers. And you see there's very nice structural organization of different types of immune cells. And that really then allows you to benchmark and test your assay and see how good it works. So we basically put this together, and the first thing that we did is run it on a human tonsil, and this is essentially then the first data point that you always get from these assays, a microscopy image of an, for example here, H&E stained tonsil. And this can be as high resolution as you want. You can also then send it to a pathologist if you want for annotation. And then we ran a 25-plex antibody panel on this and transcriptome wide probes. And this 25-plex panel contained intra and extracellular markers. So the first thing that we then look at is how does the protein data cluster? And then we project it back onto the XY coordinates. And what you see here is very nicely that these follicles essentially have individual colors. So that, they have this one color. That means they're all transcriptionally very similar as expected. And you see all the different layers surrounding those follicles, and the tonsil having their own sort of cluster identification. And when you look at the whole transcriptome data, it looks very, very similar. So we're really happy to see that the whole transcriptome and this antibody panel is actually able to divide up that tonsil into similar morphological structures. But of course, this panel was designed to be working well on a tonsil. We were hoping actually to see these different structures lighting up. I'll just show you now a couple of examples for marker genes, how this looks like. So I'm just showing proteins right now. So pancytokeratin is a marker for epithelial layer, and you see the epithelial layer here in the tonsil very well in the H&E image, and we also see this in the data. So pancytokeratin is really lining the epithelial and layer and the crypts of the tonsil. Pancytokeratin is obviously also a very interesting marker that's a lot used for tumor biology, because tumors start expressing a lot of keratins as well. Then smooth muscle actin is a marker which essentially highlights fibroblasts, but also smooth muscle cells, and we see this here. So this is mesenchymal layer of the tonsil, where there's also blood vessels visible under the microscope. You see that smooth muscle actin is also measured on protein level. Vimentin, which is also a mesenchymal marker, so it's also a fibroblast marker, is also seen in these areas here, where you would expect, mostly mesenchymal cells. Immune cells are mostly these regions here. Then we have a CD3E, which is a T cell marker. It's in the areas that are surrounding the follicles, and that's also as expected. And then PAX5, which is a very prominent B cell marker, is really within the follicles itself. And then we have also a monocyte marker here, which allows you to see different types of monocytes, and then you see this also that they are surrounding essentially the follicles in the zone where the T cells are as well. So one of the things that we played with in the beginning when we optimized this is also isotype controls, because we wanted to know, do we get a lot of backgrounds when we have isotype controls? Can we see certain regions in the tissue that are very sticky? Do we need to use isotype controls for normalization or for background correction? And I'm just showing you 2 isotype controls here, and then also be mindful of the scales here because they're very different compared to the real markers. And you see that there's more like a random noise on top of the tissue, and that's sort of what we are also hoping to see, that there's no region in the tissue that's highly, spatially enriched for these for these controls, because then you wouldn't be able to trust those data sets so much. Then one of the next things that we did is obviously compare the assay to a gold standard, which is in this case immunohistochemistry. So we took a few sections of tonsil on standard glass slides and then analyzed consecutive sections of that in this assay, and stained them, for example, for CD3E in this case, which is for T cells, where you would expect it to be, in this regions here, which is surrounding the follicles. And you see this, that might be a little bit hard to see on the screen, but the brown areas here, which is essentially the stain for CD3 and IHC, is basically also surrounding the follicles as we see it in our data. PAX5, which should be mutually exclusive with CD3E, is enriched mainly in the follicles, and that's also what we see in the data. So the follicles are lighting up. So then, one of the things that we were really interested in the beginning is to understand how does the data actually compare the sensitivity on RNA if we run protein plus RNA or if we run RNA alone. Here you see 2 different tissues that we used. I'll show these 2 in the following slides. So we ran this on ovarian cancer with an antibody panel and with the on-market Visium FFPE kit. And we ran it on a breast cancer with an antibody panel and with an on-market FFPE kit. And what you can see is, the sensitivity, when this is reads per spot and then how many UMIs were captured. And you see that the sensitivity is very similar between these 2. And sometimes we even see a little bit better sensitivity because we're able to optimize the assay a little bit. So now I'll just show you, a real use case for this in a tumor sample. So this is an H&E stained ovarian cancer, and the first data point that you get again from this assay is this H&E image. And in this case, we also sent this image to a pathologist for annotation. So the pathologist essentially annotated the large carcinoma region here, so this whole area here, and then a small carcinoma here, which seems to be invading the blood vessels. So there's the blood vessels here everywhere visible surrounding it. And this small carcinoma seems to be either invading it or seems to be in between. And then the pathologist also saw immune cells in the stain. So these immune cells seem to be surrounding here the tumor and also seem to be infiltrating the tumor a little bit on the small carcinoma. And then obviously in red is the blood vessels. So we, on this very same section that we analyzed first by H&E and then got analyzed by the pathologist, we also ran a panel of 25 antibodies and the whole transcriptome-wide probes. And now basically the first thing what we then always look at is how does the RNA cluster and how does the protein cluster on this, on this section. And so for protein, you see there's a lot of more heterogeneity there than you would have maybe thought to see, or than maybe at least that the pathologist was able to see on protein, and on RNA, it looks very similar. So, and most interestingly, the small carcinoma seems to be quite different transcriptomically than the large carcinoma region. You also see this on the protein side of things. So now we want to characterize this carcinoma, either with RNA and protein. We try to understand, which one works better, or what helps, more in this case, the RNA and the protein. And if you look for structural markers, for large regions, for example, for smooth muscle cells here that highlight blood vessels, the RNA and the protein would have given us essentially a similar conclusion. So we see that the red blood vessels and maybe also fibroblasts that are expressing ACTA2 are sort of in this area here, which was also identified by the pathologist to be the area where the blood vessels are. And that's similar for also other markers that are very broad. So in this case, PCNA, which is very oftentimes used replicating cell marker. On RNA or on protein, the data almost looks indistinguishable. And Vimentin, which is labeling mesenchymal cells, on RNA or on protein, we would have had similar conclusions looking at this data. But this is very different when we look at protein. So when we look at immune cells. So here, CD8, which is a marker for cytotoxic T cells, at transcript level, you don't really see it so well. And that's what I talked about earlier, because this is oftentimes, very lowly expressed, and those cells are obviously also very small. But on protein level, we see this, very, very strongly enriched in the small carcinoma region, and then also surrounding a large carcinoma, sort of where the pathologist also annotated the immune cells. These are two more markers for different types of immune cells. So PAX5 is labeling B cells, and CD11C is for monocytes or dendritic cells. And you see PAX5 on transcript level, or CD11C on transcript level is not really conclusive in this case. But if you look at the protein levels, it's very clear where those cells are located. There in those regions here that were also identified by the pathologists as being infiltrated by immune cells. So we'll just dive a little bit deeper and see what else is there, and really to characterize the microenvironment of the infiltrating lymphocytes here in this tumor. And we have different markers for T cells. We have this pan T cell marker CD3, cytotoxic T cell marker, helper T cell marker. We have different B cell markers, and then we have different markers for monocytes and macrophages. And overall, you see that the large carcinoma seems to be really less infiltrated by immune cells compared to this small carcinoma here. But there's really something different ongoing. I'll just blow this up a little bit here with CD3E. So you really see that the small region here is very different compared to the large region. And we validated this by immunohistochemistry, just to be sure that this is really, really real what we're seeing. And this is now a section that's not so much more consecutive anymore, but it's on the same block. It's a few sections down. That's why it looks a little bit different. But you see that this region here, there's a lot of T cells, versus here it's very devoid of T cells. And now, of course, what we have here is not only the RNA data, but obviously we have also the entire transcriptome and measured behind it. And so what you can do is you can basically divide up these two areas and just do, for example, a differential expression analysis and ask what other genes are differentially expressed between these two different areas. And is there anything else interesting going on that maybe don't capture with the protein? So you do a very basic differential expression analysis. This can be done in loop in this case. And there are several genes that are significantly up or down regulated between these two different regions. And if you just look at it by eye, one of the things that pops out is this IMPG2, which is known to be sort of involved in ovarian cancers, but it's not really sure what it actually does. But it seems to be really highly upregulated in this region here. And then the other region in the tumor, so the smaller region, has much more immune regulatory genes expressed. So that's probably also coming from all these immune cells that are infiltrating here. So that's sort of the first quick summary of this. So this assay is going to allow you to do, get essentially three readouts from a tissue section. You will be able to do a microscopy image, either with immunofluorescence or with H&E staining. And then from this very same tissue section, you'll be able to get RNA and protein expression. Again, one of the main differences here is that we were launching together with a validated panel. And this will contain dozens of markers of intra and extracellular targets. And of course, I talked about this H&E and IF stains. One other capability that we're launching with is these large capture area slides. Now I'll talk a little bit more about this in the following slides. And that's really because we heard from many customers that the 6.5 by 6.5 millimeter surface is really too small for many tissues, and oftentimes you want to have something significantly larger. So that's essentially breast cancer now run on the small capture area slide, so 6.5 by 6.5 millimeters. And again, the first thing what we did is we essentially sent this H&E image to a pathologist for annotation. The pathologist was able to see ductal carcinomas in situ here, and then also some normal glands and blood vessels, and the immune cells essentially everywhere speckled around here. And again, we did protein analysis with the 25-plex panel and whole transcriptome, and then clustered it back onto the XY coordinates. And you see that there's a lot of heterogeneity here, and this region here, for example, is sort of highlighting specifically. And I'm just going to show now two markers, for example, CD8, which is highlighting cytotoxic T cells, and HLA-DR. And so there's infiltration of cytotoxic T cells in this tumor, and HLA-DR is a marker for antigen-presenting cells, and it really seems to be that there's an active antigen antigen-presenting response going on here in this tumor. But obviously, this is actually a punch of a much larger tumor that we originally had in FFPE, and now with the large capture area slides, we were actually able to run the original the mother block of where this punch was coming from, and that's what you see here. So this is actually the large capture area slide, the 11 by 11 millimeters, and this was the punch that we have taken before. So that's this area that you see here. And you see that on the large capture area sites, you can see significantly more, and there's significantly more to visualize and study in this large tumor now. And this also runs with the protein and whole transcriptome assay. So that's protein clustering and whole transcriptome clustering here. And you see that there's just much more richness of data that you can deduce and from these large capture area slides. Just to show you a couple of markers so that this actually works. So that we have different monocyte and macrophage markers here, and you see that this area here, for example, is more infiltrated by CD68 positive cells, which are essentially the classical macrophages. And then here there seem to be also so there are no classical macrophages here, but here we see a lot of dendritic cells infiltrating. And then T cells and B cells, you can also see where they're located in this tissue section. Cool. So that was actually pretty fast. So I guess there's a lot of time for questions. This is the summary. So obviously RNA has been proven to be really powerful, for biomarker discovery, for atlasing, for really understanding the heterogeneity in tissues. But for immune cells, particularly for studying really cell states and activation cascades, protein is really not the entire. So RNA is not the entire story, and protein really will be added value to this. And the gene expression and protein assay that will be launched in the middle of this year will allow you to really merge these two worlds together. So you will get the whole transcriptome readout, high-plex protein, and then also the morphological image of the same tissue section.